In this video, we'll cover certain concepts involved when molecules are trying to move across membranes. Ions and small molecules in a solution have some thermal energy and therefore are in constant random motion within the solution. This random movement is called diffusion. Sometimes these small molecules and ions move across or diffuse across membranes, and this movement is called passive transport. This kind of transport does not require any input of energy. On the other hand, another kind of transport called active transport requires energy to move certain substances across the membrane. Now, why do molecules diffuse across cellular membranes? This is because the concentrations of solutes across membranes can be different on the outside compared to the inside. This results in a concentration gradient and enables these ions and molecules to move across cellular membranes from an area of higher concentration to an area of lower concentration. This process continues until equilibrium is reached, meaning that the concentrations of solutes on either side of the membrane are equal. Note that molecules are still moving randomly within the solution, but there is no net movement of the solutes from one side of a membrane to the other. Take, for example, two solutes that are separated by a selectively permeable membrane, solute 1 and solute 2. There's a higher concentration of solute 1 on the left side of the membrane, and there's a higher concentration of solute 2 on the right side of the membrane. Since the membrane is permeable to both solute 1 and 2, both of these solutes will move down their concentration gradient from an area of higher concentration, which is the left side for solute 1, to an area of lower concentration, which is the right side, until the concentration of these solutes is equal on both sides of the membrane. Note that this is because the membrane is permeable both to solute 1 and to solute 2. So far we've seen that molecules and ions are constantly in motion because they possess thermal energy. In this simulation, there are two kinds of molecules, blue and orange. Now let's see what happens when we put a trace on one kind of molecule. You can see that the molecule is randomly in motion and its path is being directed by collision with other molecules that are present in solution or with the sides of the container that is holding the solution. So there's no particular pattern to the motion of that molecule. Alright, so now we're going to eliminate the trace walk and add a membrane to this container. Now this membrane does not allow any molecules to move across it, so both the blue and orange molecules are colliding with it and then changing direction and are being unable to cross this particular membrane. Right now it's not permeable to either blue or orange molecules. Now let's make it permeable to the blue molecules. And let's make it permeable such that these blue molecules are able to move to only one side, in only one direction. Now you can see that over time, the blue molecules are moving from the left to the right-hand side of the membrane because they can only move in one direction. So now they all collect on the right-hand side. And if we make the membrane such that the blue molecules are able to move across both sides of the membrane, then these blue molecules will spread out and move and diffuse across the membrane till their concentration reaches equilibrium. A few other variables we can add to this system is increasing the temperature. So if we're putting in more energy into the system, these molecules move faster because they have more energy. Their motion increases. And if the membrane is permeable to the blue molecules, you're going to see them cross the membrane more quickly. You can also change the mass of molecules, and in particular the orange ones. And you can see that higher mass means less speed of motion. So the orange molecules are moving more slowly than the blue molecules because they're larger, which makes sense. And now if we increase the temperature, they will start moving faster because they have more energy, but again, not as fast as the blue molecules. Decreasing the temperature of the system results in a slower movement of molecules. The specific movement of water across lipid bilayers is a special case of diffusion called osmosis. Water is special because it moves from regions of low solute concentration to regions of high solute concentration. So the thing to keep in mind here is that water always follows solute. As a result, this movement of water dilutes the higher concentration, equalizing the concentration of a solute on both sides of a lipid bilayer. Osmosis only occurs across selectively permeable membranes. So if solutes are not able to move across a lipid bilayer, water moves to dilute solute concentrations and bring about equilibrium. In this image, we can see that a selectively permeable lipid bilayer separates two compartments in a beaker, and there are two different solute concentrations on either side. The left side has a lower concentration of the solute than the right side, and the membrane does not allow this solute to pass through. Since the solute molecules can't cross the membrane to reach equilibrium, water moves to dilute the higher concentration of the solute and brings about equilibrium in the concentrations of the solute on both sides of the membrane. This movement of water is called osmosis. The concentrations of solutes inside and outside a cell can be different. If an outside solution has a higher concentration relative to the inside of a cell, the solution with the higher concentration of solute is called hypertonic, whereas a solution that has a lower concentration relative to the inside of a cell is called hypotonic. If the solute concentrations are equal on both sides of a cell membrane, then those solutions are said to be isotonic to each other. So in the case of a hypertonic solution, water will move out of the cell by osmosis to dilute that solute, and as a result, the cell will shrink because it will now have less water on the inside. In the case of a hypotonic solution being outside a cell, water will move from the outside into the cell because the concentration of the solute in this case would be higher inside the cell, and water follows solute, so water would move inside the cell, diluting that high concentration, resulting in the swelling of the cell. And in an isotonic solution, there is no net movement of water on either sides, and the cell maintains its shape. This emphasizes the importance of maintaining different solute concentrations stable both inside as well as outside cells in biological systems. So when solute concentration is higher on the outside, water from inside the cell moves out to dilute the solute outside, resulting in shrinking of the cell, or in this case a vesicle. The inside of this vesicle in this case is said to be hypotonic relative to the outside. When the inside of the cell or a vesicle is hypertonic, meaning that it has a higher solute concentration relative to the outside, water from the outside solution moves in, leading to the swelling or even bursting of the vesicle if too much water gets in. And in the third case, if the concentrations of solute are equal on both sides, then there isn't any net movement of water. Water can still diffuse from one side to the other, but there's no net movement. And the solutions on both sides are said to be isotonic to each other. And as a result, the vesicle maintains its shape. This concludes our section on membrane transport and how molecules and ions get across membranes. At the end of this section, I'd like you to be able to respond to the 3 questions listed on this slide. Thank you.